In this video, we will learn about enzymes' mechanism of action. There are four main steps in the working of enzymes. These four steps are recognition, binding, catalysis, and release. In the first step of recognition, enzymes recognize and binds to substrate through non-covalent bond. This non-covalent bond can be hydrogen bond, ionic bond, or hydrophobic interaction. After recognition, substrate binds to the active site of enzyme and form enzyme-substrate complex. In the third step, enzymes lower the activation energy required for the reaction to occur by stabilizing the transition state of substrate. This rapidly converts the substrate into product. In the final step, the product is then released from the active site of enzyme and then enzyme is ready to bind to another substrate. These were the basic steps in the working of any enzyme. But to know how enzyme combine with the substrate and convert it into product, scientists proposed two models, which is used to describe the enzyme's mechanism of action. One is the lock and key model and other is the induced fit model. Let's learn about them in detail. Lock and key model. It was first proposed by Emil Fischer in 1894. In this model, enzyme is considered as a lock and substrate as a key. As a specific key can open a specific lock, this model works the same. Not all substrate can attach on the active site of enzyme. The active site of enzyme is rigid according to this model. It do not change its shape. This model also proves the specific nature of enzyme. Enzymes that follow this model are mostly non-regulatory enzymes. That means they do not have any check or negative feedback mechanism. Its example is lipase enzyme. Induced fit model. It was discovered by Koshland in 1958. This model proposed that the active site of enzyme is flexible. It can get altered according to the substrate. Enzymes remain inactive when substrate is not bind to it. But when substrate attached on enzyme, active site of enzyme undergo some changes and become active. These enzymes are regulatory enzymes. Its example in the body is glucokinase. It is symbolized with glove in hand.